Good afternoon, everyone, and welcome to the Department of Medicine's Grand Rounds. Please take a moment to silence your pagers and mobile devices. Grand Rounds next week will be another Chief Resident Grand Rounds presented by Dr. Sonia Abichandani and Dr. Zil Patel. Today, I have the pleasure of introducing Dr. Alan Shuldiner, John L. Whitehurst Endowed Professor in Medicine, Associate Dean for Personalized and Genomic Medicine, and Director of the Program in Personalized Medicine here at the School of Medicine. A leading national expert and researcher in personalized medicine, Dr. Shuldiner focuses on the genetics of age-related diseases, including type 2 diabetes, obesity, osteoporosis, and cardiovascular disease. He is best known for his studies involving Old Order Amish, a homogeneous population ideal for genetic studies. He leads a multidisciplinary research team that uses state-of-the-art molecular, genetic, statistical, and epidemiological methods. His group reported the first null mutation in the APOC3 gene and its association with low blood triglyceride levels and cardioprotectin, which validates treatments for hypertriglyceridemia. Most recently, his group identified a common gene variation that reduces the benefit of clopidogrel, which many cardiologists now use to individualize antiplatelet therapy. Dr. Shuldiner also serves as vice president and co-head of the Regeneron Genetics Center, a program that focuses on early gene discovery and functional genomics and facilitates drug development. He has authored more than 300 original articles in leading journals, as well as 78 reviews and book chapters. Speaking to us today on the path to precision medicine, please join me in welcoming Dr. Shuldiner to the podium.
Great. Well, thanks so much. It's really a pleasure to be here, speaking in front of the home team crowd here. Really fantastic. And also, at the same time, intimidating, because many in the audience are responsible for a lot of the great work that I'll present today. And also, really an honor and a privilege, not just to present here at Medical Grand Rounds, but in terms of co-sponsorship with the newish ICTR program. Really fantastic to see that mature into reality. And in fact, I dedicate sort of the theme of my talk today on translational medicine and translational genomics. And I think the bottom line message is to convince you all that the path from basic science and basic discovery to translation into evidence-based clinical medicine is a long path, but one in which we all aspire to and for which our patients ultimately benefit. So that's the ultimate theme here, and already disclosure, oh, what was that? Oh, I might have to, okay, yeah. Great. So you've already heard a disclosure in that in addition to my part-time appointment here at the University of Maryland, I also work for the Regeneron Genetics Center. And in fact, a lot of the work that I'll tell you about today is the product of collaboration between the University of Maryland group and the Regeneron Genetics Center. Another example of how we can facilitate translational medicine through collaboration between industry and academia. So in terms of my lecture outline today, I know that we're relatively limited for time. I may have to skip a story or two. But what I'd like to first tell you a little bit about in terms of an advertisement is to tell you about the University of Maryland's program for personalized and genomic medicine, and in particular the resources for clinical and translational research that clinical researchers and scientists, ICTR investigators can tap into for their research. So that'll be about a 5-slide advertisement. Some of you know about the program for personalized medicine and the resources, but I bet a lot of you don't, or at least are not up to date with regard to resources that are available broadly for researchers here at the University of Maryland. And then secondly, I'm going to talk about the application of human genetics for therapeutic development. And I'll provide some examples of how now high-throughput human genomics and linking through high-throughput human genomics to electronic health records from large patient populations can actually really markedly grease the wheels towards identifying new therapeutic targets, de-risking therapeutic targets, and also potentially finding new indications for therapeutic targets. You won't get away with any talk of mine without me talking about founder populations, because I think they really provide very unique opportunities for novel gene discovery that couldn't be found in millions or even hundreds of millions of patients from general populations. And so I'll tell you a little bit about some of the genomics more recent genomics and founder populations as new opportunities. And finally, what I'll get to at the end is the idea that we're moving towards now, truly implementing precision medicine, and the challenges that we come as healthcare providers as we begin to understand the human genome and as the human genomic information in each patient is now being used for patient care. And I'm going to talk a little bit about the Geisinger Health System experience, since I think they're probably 5 to 10 years ahead of other healthcare systems in terms of how they're thinking about human genomics and patient care. So the advertisement for the Program for Personalized and Genomic Medicine, the mission here is to advance discovery in genomics and other omic sciences, to accelerate translational research and implementation of discoveries into more effective and safe individualized healthcare, and to enhance training and education of future generations of healthcare providers and scientists. And the program has been in existence now for almost a decade. And as I kind of envision the program is that we're here to grease the wheels from genomic discovery to clinical research, ultimately implementation science research to build the evidence base for translation into patient care. And you can see this slide summarizes some of the activities that PPGM has been involved with across that continuum from discovery to clinical research to ultimately evidence-based translation to patient care, education, a key piece to this. And I'm going to highlight in the next 2 or 3 slides some of the major resources that faculty can tap into. One thing that's very important for translational research is to have a laboratory that is CLIA and CAP accredited so that they can do genomic testing in a way that those results can be clinically validated and also used in clinical care to actually study the effects of implementation of a particular genetic or other test into patient care. And we now have the Translational Genomics Laboratory, which is part of the large cyber core facilities here at the University of Maryland. And this facility can do CLIA validated genetic testing and can actually bring up whatever genetic tests are required for a particular study initiated from any University of Maryland investigator. You can see some of the tests that have already been brought up and which are running, some of them that are in progress, and a few of these, particularly CYP2C19 genotyping, has actually made it into standard of care as one of our first successful implementation pharmacogenetics projects here at the University of Maryland. So anyone that has need for CLIA CAP accredited genetics, the Translational Genomics Laboratory is open for business. In addition, now, several years ago, we got a large shared instrumentation grant to purchase a Hamilton BIOS automated ultra-low temperature storage system. This monster is located actually on the 6th floor bridge in HSF1. It took about 3 years to set up because they had to reinforce the floors in order to install this thing, but it's now installed and fully operational, and will accommodate up to or even over 900,000 samples. We have also automated liquid handling for blood processing and DNA isolation. We now have more than 40 current users that store their samples in this system, and it's also the home of the University of Maryland MGen Biobank, which I'll tell you a little bit about on the slide to come. And also now, as part of the biorepository resource, we have a new liquid nitrogen storage opportunities as well. So anyone that has samples that they would like to put in a safe place, be able to retrieve them readily, and do that at a very in a very efficient way. The Biorepository is open for business. Part of the Biorepository houses the University of Maryland Genetics Biobank. We call this MGen. And over the last 3 years, a number of people have been working very hard to get informed consent for University of Maryland patients across the University of Maryland medical system and also across FPI. We've now enrolled a bit over 2,500 patients. All of them are consented for very broad future research use of samples as well as data for, and also have the option for recontact for additional studies. This is a broad base of patients that have all of the common disease diagnoses or on all of the common medications, and again is open for use by any University of Maryland investigator to tap into the data as well as serum, plasma, or DNA, which is available on all of these patients. And there's also opportunities, of course, to link to the electronic health record data through the Research Harbor, and that could retrieve information from patients in a longitudinal way. So any uses or needs for 2,500 plus patients, either as controls for your study, or maybe you're interested in a particular disease, we'll have probably several hundred, if not more than 1,000 cases for things like diabetes and obesity, all the common disorders. The biobank is open for business. And finally, let me tell you a little bit about the Amish Research Clinic. This is considered an ICTR resource for clinical and translational research. We already have a large group of faculty here at the University of Maryland who have been working at the Amish Research Clinic and have been studying many different disorders and diseases and human phenotypes. The clinic is located in Lancaster, PA. It's soon celebrating its 25th year of continuous operation. You can see our wellness mobile here. And the cool thing about this is that this is a resource, again, that any University of Maryland faculty member can tap into and use for their clinical research. And you'll hear why in some slides to come the Amish are quite opportune for these kinds of research. In addition, layered over the recruitment of over 6,500 Amish individuals over 25 years, we also have whole exome and or whole genome sequence data on the vast majority of these individuals, hundreds of phenotypes and traits, also a wonderful browser called the Oasis browser, founded by Jim Perry here at the University of Maryland, which is a very user-friendly database in which one can query genotype-phenotype relations in this vast genetic and phenotype database. So if you have a gene of interest or a phenotype of interest, you can very readily have access to Oasis and ask very readily the questions about whether your gene of interest is connected to any of the phenotypes that you may have interest in. And if not, we can actually call back many of these Amish individuals for prospective callback studies. Obviously, this information can serve as preliminary data for grants to get new funding for these callback studies. So a great resource available again to University of Maryland investigators. And I'm not going to go through this whole slide, but the Program for Personalized Genomic Medicine also provides clinical genetics. I should say doesn't provide, but helps assist and facilitate clinical genetic services. This is showing some of the services for adult clinical genetics. I'll point out a new pharmacogenetics service led by Amber Beidelschiess, which has also enabled some of our implementation programs in pharmacogenetics. Okay, so that was my advertisement. Let me now move into the next phase of my talk, and that is to talk a little bit about how human genetics is being used for therapeutic development. And the challenges that the biotech and the pharmaceutical industry faces to date is the fact that we are very inefficient at finding therapeutic targets that turn out to be effective in human beings. And this is despite huge expenditures in research and development in the pharmaceutical industry, also, of course, by NIH. And despite all this research and development, more than 90% of molecules that actually go into humans fail to demonstrate safety or efficacy. And most of these failures fail really pretty late in the game when hundreds of millions of dollars have been spent on developing that therapeutic target and that therapeutic agents. A lot of it due to lack of efficacy, also due to toxicity. And this has led to the conclusion that whatever we're doing is not right, that these preclinical models, whether it be mice or other preclinical models that we identify these therapeutic targets in, are just not good predictors of efficacy or clinical benefit in humans. And this has led to the idea that compounds that are supported by human genetic evidence are substantially more likely to succeed in humans. So the idea here is that, and there are now several examples of this. I'm showing two on this slide. Many of you know about the PCSK9 story where back in 2003, rare families were identified that had gain-of-function mutations in PCSK9, and these individuals had familial hypercholesterolemia. Not long after that, population studies actually identified rare loss-of-function variants in this same gene, and these individuals had very low LDL cholesterol levels, and they were protected from coronary heart disease. And this was essentially a genetic validation of PCSK9. It's a drug target for LDL lowering, and now two FDA-approved medications showing efficacy for LDL lowering, and also emerging studies on cardioprotection as well. The second example I love to show is this one down here, in that this initial discovery was made by Tony Pollan here at the University of Maryland, the first null mutation in ApoC3 in the Amish. This was associated with very low triglyceride levels and was cardioprotective. Not long after, two large population studies essentially showing the same, and now several pharma and biotech companies now have in various stages of clinical trials ApoC3 inhibitors, and these are proving to be quite effective for triglyceride lowering and we hope as well cardio protection. Many other examples as well. So we can now cast our net across the entire human genome and ask how many of these genetic experiments of nature exist in the billions of humans that exist across this planet to be able to inform us about what are the correct and likely most efficacious targets in humans for new medication design. And this is what's being done now really pretty much across the industry. So we're applying human genetics for target discovery, to identify new targets and pathways. We're using human genetics to identify new indications. We're using human genetics to de-risk various targets. And I'm going to provide examples of each of these three just to make this real for you. And of course, there's the area of pharmacogenetics where we can use genetics or other biomarkers to stratify patient populations and be a little bit smarter about who we prescribe these medications to. And you can see we've turned this entire therapeutic target development paradigm on its head, in that then we will then use preclinical models to dive deeper into the biology only after the human genetics has provided that validation step that I just mentioned. So let me tell you a little bit about one story. And this story starts with a collaboration between the Geisinger Health System and Regeneron Genetics Center. We're working with Geisinger to sequence at least 250,000 of their patients linked to de-identified electronic health record data, ultimately to make some of these genotype-phenotype associations that inform human biology and ultimately the identification of therapeutic targets. So they're located in central Pennsylvania. They service about 1.6 million participants, predominantly European Caucasian. They were very early adopters of the electronic health record, so they have very good longitudinal data in their patients. And as I mentioned, they have a very large biorepository. They have over 150 patients consented into their MyCode Discover cohort. A bit over 90,000 of them have been sequenced, exome sequenced, and we now have an amazing data set to begin to embark upon these analyses. So I'm going to summarize for you some of the pertinent results of a publication that came out now a couple of years ago describing what we learned about the first 50,000 Geisinger patients that were sequenced as part of this collaboration. For one thing, this is what the electronic health record data looks like. Really important to be able to extract high-quality electronic health record data in order to do this scale of research. And Geisinger is second to none at this. You can see that they have a very stable patient population. Over 400,000 of their patients have 10 years or more of longitudinal electronic health record data in the Geisinger EHR. You can see also across the system that there are millions of laboratory values that can be analyzed and hundreds of thousands of individuals with some of the most prevalent and common diseases that we're interested in understanding better from a genetics perspective and ultimately to make better medications for. And so that's what we know about the electronic health record and what we can use for discovery research. What do we know about their exomes? So this is aggregate data from the first 50,000 exomes that were sequenced from these Geisinger patients. For one thing, you can see that we identified a bit over 4 million variants. And for those of you who don't know what exomes are, that's about 30 million base pairs of essentially the coding regions of all of the genes in the genome. So it's only about 2% of the whole genome, but it's sort of the business end of the genome and the part of the genome that we would like to understand most in order to understand human biology and ultimately find new therapeutic targets. So we have about 4 million variants in that region of the genome, the exome. You can see that the vast majority of these variants are very rare. They have a frequency of less than 1%. So this is why you need very large patient populations to be able to make sense of this genetic data. And you can see that we have over almost 200,000 variants that would predict a loss of function of that protein. These are essentially individuals who are heterozygous or homozygous for a human knockout. Just like you'd make a mouse knockout, we can study human knockouts with human genetics. And this is what we're essentially up to. If you boil this down to the individual level, each individual on average is heterozygous for a loss of function variant in about 21 of their 20 or 25,000 or so genes. And each of us is a homozygous essentially knockout for about one gene in our genome. And so this is what makes everyone in this room a little bit different from the person sitting next to you, in that you have different sets of genes that have different variants, and even some of them total knockouts in function. And if you look at this over the 50,000 exomes that were sequenced, we have over 17,000 genes of the 25,000 or so that we all have, in which we have at least one individual who is heterozygous for a loss of function variant. And we have 7% of the genome, over 1,300 genes, in which we have at least one individual who is a homozygous knockout. So now we can go back and ask what diseases, what problems do these individuals have, what do the electronic health records look like, and really begin to understand what these genes are doing in human beings. So as a proof of principle, now with all of that data, the phenotype data and the genotype data, we can sort of retrospectively ask, with this data set, could we have predicted what the efficacious therapeutic targets are for LDL lowering? So these are drugs that are already either FDA approved or well on their way. We know what their target is. We know that they're effective and they're pretty safe. And the question is now with our genetics database, could we have predicted that these would have been effective targets? And the bottom line here is that 8 out of the 11 lipid therapy targets that harbored loss of function variants showed nominally significant or directionally consistent clinical associations that recapitulate that drug's effects. So the examples I'll show you quickly here, ApoB, mipramycin is an antagonist. We could have predicted genetically that this would have been a good target because those individuals who have loss of function variants in ApoB have lower LDL cholesterol levels, recapitulating essentially what the FDA-approved drug actually does effectively and safely. PCSK9 as well, now 2 FDA-approved drugs, would have predicted with very high statistical significance that knocking out function of that gene would result in LDL lowering. And again, Tony and my favorites, ApoC3 would predict genetically this to be a good target for triglyceride lowering because individuals with loss of function variants in this gene have much lower triglyceride levels. So that's retrospectively a proof of principle experiment. Now, what can we do prospectively to identify new therapeutic targets for lipid lowering? And the story I'm going to tell you now is of angiopoietin-like 4 and some of the genetic studies that have validated this as a very promising therapeutic target, new therapeutic target for triglyceride lowering and protection from coronary artery disease. So this is a complicated slide, but the bottom line is that when triglycerides come in through the intestine, they come in and are on these chylomicron particles. And in the endothelial cells, in the capillaries, is lipoprotein lipase, and lipoprotein lipase is responsible essentially for hydrolyzing these triglycerides into free fatty acids that can then be used by muscle and adipose tissue and or taken back up by the liver for storage. So LPL is an important molecule involved in triglyceride homeostasis, and it's one might imagine is very tightly regulated. And in fact, there are multiple inhibitors of LPL, ApoC3 among them, but also these angiopoietin-like molecules, angiopoietin-like 4, also angiopoietin-like 3, that inhibit LPL, and again, in a very regulated way. So the point being here is that one could imagine that if one would inhibit angiopoietin-like 4, you would then increase LPL activity and ultimately potentially decrease triglyceride levels in the circulation. So how can we test this hypothesis genetically? In those 50,000 or so Geisinger patients, we were able to identify 80 some odd individuals who had loss of function variants in angiopoietin-like 4. And these are the loss of function variants that you see. All of them are rare, but you can kind of add them up in aggregate. And you can see when you do that, that those that have loss of function variants in angiopoietin-like 4, on average, have lower triglyceride levels. Cholesterol is not that much different. They tend to have higher HDL levels. LDL, glucose, BMI are not all that much different. So in the table that is summarized, here are the, it's hard to see from here, individuals that have loss of function variants, and you can see they have significantly lower triglyceride levels. There's also a common loss of function variant, E40K. You can see that there are many of them, and similarly, they have significantly lower triglyceride levels than those with 2 normal copies of angiopoietin-like 4. So again, this points to the idea that if one could inhibit angiopoietin-like 4, potentially with a therapeutic, that would lower triglyceride levels. And in addition, you can look to see whether these individuals are protected from coronary artery disease, and you can see again in these Geisinger patients that that have these variants, they have a lower risk, a lower odds ratio of having coronary artery disease, again suggesting that they're protected. And in fact, angiopoietin-like 4 monoclonal inactivating antibodies are available, and these are some of the studies that were done in the quote-unquote preclinical models. You can see when this antibody against angiopoietin-like 4 is administered to mice, homozygous for human angiopoietin-like 4, it has potent triglyceride lowering properties. Here are rhesus monkeys, again, potent triglyceride lowering, cynomolgus monkeys, potent triglyceride lowering. One of these rhesus monkeys with extremely high triglyceride levels, one dose of the antibody lowered the triglyceride levels literally to 0. So we have human genetic evidence. Now the preclinical models are validating this evidence. But one big problem is that some of the mice and one of the monkeys actually developed abdominal lymphadenopathy and chylous ascites. And in fact, one of the monkeys actually died. So this would kill any clinical program right away, obviously. But we can go back to the human genetics and ask the question, is this risk relevant in human beings? So if you go back to these individuals who carry these loss of function variants, including homozygotes, these are individuals who are recapitulating the drug, if given, and you go back to the electronic health records and ask whether they have any problems with lymphadenopathy, chylosocites, abdominal pain, et cetera. So these 17 homozygotes, among them over the 10 years that they were Geisinger patients, 5 of them actually had CT scans of their abdomens. And 4 out of 5 of the reports had explicit mention of normal abdominal lymphatics. 1 out of 5 of them had no mention of lymphatic abnormalities. Chart reviews essentially showed no greater increases in any of these issues that we were worried about in the preclinical models. So this is a way to sort of de-risk this particular therapeutic target and maybe move it on into humans, despite the fact that preclinical models would have suggested otherwise. And then my example of new indications, once you have genetic data linked to the entire medical phenome, you can actually ask whether loss of function variants are associated with any other phenotypes that you might not have anticipated. And as it turns out, when we went ahead and did that, these same individuals that had loss of function variants in angiopoietin-like 4, they actually were protected from type 2 diabetes. And I won't go through all of the data here, but this required a very large sample size, a meta-analysis, ultimately showing that individuals with these loss of function variants were protected from diabetes. And this to us suggests maybe a new indication for angiopoietin-like 4 inhibition, that is to treat and/or protect from type 2 diabetes. So that's a good example of how human genetics is applied to validating a drug target in humans, de-risking it, and also finding a new indication. I'm not going to have time to tell you at all about this exciting story, but again, through a very similar approach, we've identified a protein truncating variant in a new gene called HSD17B13. This loss of function variant appears to protect from chronic liver disease, pretty much all chronic liver disease, including non-alcoholic fatty liver disease, and now a very active program to try to find the substrates for HSD17B13, understand the biology, and ultimately potentially advance this as a therapeutic target for which genetics would predict inhibition would protect from chronic liver disease. Okay, so I have about 15 or so minutes left. Let me move on to the founder populations and the opportunities for novel gene discoveries to, again, grease that pipeline to translational medicine. Many of you know that founder populations are unique in that there are a limited number of individuals that created that particular population. And then through isolation, we have a population that is much more genetically homogeneous than the more general population. And through the generations, there is the opportunity for rare variants that get into the population through the founders to actually increase or decrease in frequency. This is called genetic drift. And when they increase in frequency, of course, it provides the opportunity to identify these rare variants much more readily in these founder populations than it would be in more general populations. In addition, some of these founder populations also practice endogamy, and this allows these drifted variants to become present in their homozygous form. And if the phenotype is recessive, you can identify them much more readily in founder populations. And finally, founder populations have typically a more homogeneous lifestyle, which reduces some of the confounding influences of genetics. So just as the Amish that we've worked with for many years are one founder population, one human genetic experiment of nature that has occurred in Lancaster, PA, and other parts of the Midwest, there are thousands of founder populations throughout the world. They live on islands, they live on hilltops, they live in valleys. Many of them live among us in terms of being religious isolates, and this provides essentially thousands of independent experiments of nature to understand human genetics through studies of these founder populations. And one of the things that I've been really excited about over the last several years is the establishment of what we call the DRIFT Consortium for Discovery, Research, and Founder Populations. We now have accumulated more than 17 academic collaborators, over 120,000 samples from more than 80 distinct founder populations from around the world. And this is now going to be a fantastic resource for novel gene discovery. So the examples I'll provide now are ones that we have come across in studies of the old order Amish. And as I've mentioned, these studies have been going on for now about 25 years. And of course, I don't have time in 5 minutes to summarize 25 years of research, but this slide kind of shows you kind of what we have learned, or some of the salient points of what we've learned. I've told you already about APOC3, the fact that 1 in 25 Amish carry this drifted variant, and that seems to be a good thing in those carriers. You heard about a variant in CYP2C19 as a predictor of clopidogrel response, and some of the initial discoveries were made in our studies of the Amish. I didn't say much about familial hypercholesterolemia, but through genetic drift, familial hypercholesterolemia is extremely common in the Amish. 1 out of every 8 Amish have familial hypercholesterolemia. In the general population, it's about 1 in 250. Real opportunities to really understand FH in a much deeper way by studying the Amish. LIPE, this is work of Colleen Damcott and colleagues, a new variant, one that really, as far as we know, probably doesn't exist anywhere else outside of the Amish, or if it does, it's very rare. And this is a new form of lipodystrophy in its homozygous form, and also increases risk of typical type 2 diabetes in its heterozygous form. We work very closely with the Clinic for Special Children in Strasbourg, PA. Kevin Strauss and his colleagues have been doing a lot of exome sequencing of children with very rare disorders, some autosomal recessive, some de novos, but many new genes discovered to be linked to these rare genetic disorders. And with whole exome sequence now available in over 6,200 Amish, a whole host of new discoveries that are emerging. And the two that I'm going to try to have time to tell you a little bit about is a novel gene for serum lipids and thrombosis, potentially other indications, and also a drifted allele in KCNQ1 that makes long QT syndrome and sudden death a major population health problem in Lancaster. Okay, so this is the work of May Montasser and many people here in the room. It's a genome-wide association study with exome sequence data, and you can see association on the y-axis in terms of p-values for association with LDL cholesterol level, tens of thousands of variants in the exome here. You can see that ApoB variant was not difficult to identify. That was previously known. But you can see there are some new signals that are coming up here that have not been found before. And one of them is a variant on chromosome 9 that turns out to be very enriched, associated with LDL cholesterol, that intends to be very enriched in the Amish. Frequency is 6% in the Amish. You can see in Geisinger, very uncommon. Over 200,000 individuals from the NOMAD database, only 2 individuals that carry this variant. So this association would not have been detected anywhere else except for a population in which it was particularly enriched. And as it turns out, this is a missense mutation in a gene called B4GALT1. You can see the very strong associations with lower total and LDL cholesterol levels, and very robust statistical evidence that this is a real finding. I should also point out associations with lower levels of fibrinogen. Many of you know that fibrinogen is an independent risk factor for coronary heart disease, and this suggests that maybe this is a potential therapeutic target that could protect from cardiovascular disease, not just from LDL lowering, but also maybe from inhibition of thrombosis. So what is B4-GALT1? It turns out that this is a galactosyl transferase. So you've got to go back to your glycoprotein chemistry and organic chemistry or biochemistry to remember this stuff. But B4-GALT1 essentially attaches a galactose onto the growing chain of glycoproteins, ultimately capped by sialic acid, but you can see in yellow here the galactoses. And that's what this enzyme does. It adds galactose to the glycan chains of glycoproteins. And as all of you know, there are thousands and thousands of glycoproteins in our bodies. It turns out that this particular missense variant, serine for asparagine, is very close to the active site of this enzyme and would be expected to decrease or alter the activity of the enzyme. And in fact, that's what we've been able to demonstrate. These are mass spec of serum glycoproteins, and you can actually characterize all of these partially processed and fully processed glycans in these glycoproteins. And you can see that in individuals who are homozygous for the serine allele, their plasma proteins have significantly less galactose and significantly less sialic acid. And that's true for global glycans. If you look specifically at fibrinogen, you can see a marked lowering in galactose on the glycan chains of fibrinogen, and same thing with ApoB, and in addition, lower ApoB levels. So I'm showing you fibrinogen and ApoB for obvious reasons, because that's connecting essentially now the biochemistry to the clinical phenotypes. And I can't say we know everything about this variant at this point in time, but as we begin to uncover deeper the biology of glycosylation, and in particular the role of B4-GALT1, we're confident that we will identify potentially new pathways and potentially new therapeutic targets. Naran Zagul, with her zebrafish model here at University of Maryland, was able to knock down B4-GALT1 in zebrafish and measure LDL cholesterol levels. You can see that these B4-GALT1 knockdown zebrafish have lower LDL cholesterols that can be rescued with the wild-type B4-GALT1 and only partially rescued with the mutant B4-GALT1. And in experiments at Regeneron in collaboration with the group here, mouse knockdown of B4-GALT1 in mouse liver, specifically, again, recapitulates the phenotype, lowers LDL cholesterol in these mice. So glycosylation is a global, a very ubiquitous phenomenon. While it appears to affect LDL cholesterol and fibrinogen, it likely affects many other processes, and in fact, B4-GALT1 and this process has been implicated in immune function, also inflammation, particularly in the colon, to some extent in reproduction as well, and maybe even in cancer. So we hope to understand deeper the biology of B4-GALT1 that might shed light not only on the phenotypes we initially identified, but potentially more broadly. Okay, I'm doing pretty well for time, maybe 5 more minutes here. This next story is really a very cool story. Through exome sequencing of over 6,000 Amish individuals, we came across a variant in KCNQ1 which causes long QT syndrome. This is just an introductory slide for those of you who don't know about long QT syndrome, or not as much as you might need to know to interpret the couple of slides to follow. It's pretty rare. I mean, it affects only about 1 in 7,000 people, responsible for about 3,500 deaths in the US. It's highest risk for cardiac events, typically in youth. Emerging evidence that maybe 1 in 10 crib deaths may be due to mutations in genes that cause long QT syndrome. The most common mutation is in mutations in a gene called KCNQ1, which encodes a cardiac potassium channel. It's autosomal dominant. There are other genes that are involved. And one of the important things about long QT syndrome is that it's actionable. A beta blocker and also avoiding provocations of further QT prolongation can actually prevent sudden death and syncope in individuals carrying these variants. So again, this is the work of the University of Maryland group and a major effort by Liz Streeton and Amber Beidelschiess to follow up on this. This is the actual data that brought this finding to light. We found in the exome sequence data a variant in KCNQ1, threonine 2,2,4-methionine, that had a frequency of about 1% in the Amish. 1 out of every 40 Amish carries this variant. And it's highly associated with QT interval, p-values of 10 to the minus 20th. The effect size is about a 20 millisecond prolongation in QTc in individuals who carry this variant. It turns out that it's very rare in the general population. In NOMAD, the frequency was 1 in about 250,000 individuals, so 1% in the Amish is quite high. This was a variant that was characterized previously as a variant of unknown significance, in that there was one individual that had this variant in a collection of over 2,500 patients with long QT syndrome, but no further evidence that this was indeed pathogenic until the findings here that were uncovered by the University of Maryland group here. So in chart review of these 120 plus individuals who carry this variant in the 6,000 that were studied, 85% of them had an abnormal QTc, suggesting that indeed this is a pathogenic variant, and of course this has important implications to the Amish community. So really cutting to the chase, once we felt that this was potentially an actionable variant important for population health in the Amish, we got IRB approval through a process to offer return of results to our Amish research participants. And I'm sure Liz, when she gives her grand rounds, maybe next month or next year or whatever, will go into this in much greater detail. But we're really creating new ground here in that there weren't really explicit, it wasn't explicit in many of the consents that we would be returning results. So we had to make a proactive decision ethically about what was right and what we should be doing going forward. And hats off to our IRB. They agreed that this was something that we as researchers had an ethical obligation to return, and we went through a mechanism in which I think we covered all the bases. Anyway, Liz went back and visited virtually every one of these 120 some odd individuals who wanted to know something about actionable results, and you can see the information that was uncovered here. 83% of the carriers had an abnormal QTc. That was on a repeat electrocardiogram that was done in the home. Non-vasovagal syncope through family history and medical history was much greater in the carriers of this variant. 40% of the carriers had a family history of a first or second degree relative that had sudden death, compared to only 8% of non-carriers. And some of these non-carriers might have had family members who were carriers. So a marked increase in syncope and sudden death in these carriers. And I think, you know, these pedigrees tell the whole story, I think. And again, these are ones that were constructed by Liz and Amber Beidelschiess and colleagues. You can see these are families that through family history, one might have just thought, you know, we're just having a lot of bad luck. But we now know it's due to segregation of this pathogenic variant in KCNQ1. Children dying early at early ages, stillbirths as well that I think might very well be homozygotes for the KCNQ1 mutation. And again, all of this actionable with treatment with a beta blocker and some advice. So this has huge implications. 1 out of 40 Amish carry this variant. There's also the opportunity for cascade screening in offspring of research subjects. So these are individuals we have never even seen as research subjects, but in that they're related to research subjects, this finding has important clinical implications. And we're now putting in place population-wide screening, cascade screening, tracking clinical outcomes, and also evaluating the ethical, legal, and other implications of the finding. So the last three slides, fast forward a little bit and think about now what a place like Geisinger could be doing with exome sequencing data on all of their patients. Certainly, I showed you how useful it is for research, but how are they going to take this forward into real-world settings and ultimately utilize this to improve the health of their patients? Well, as it turns out, the Geisinger MyCode program anticipated the idea that they might be wanting to return results to their patients at some point in the future, and they have now a process in which they can confirm the research finding, and then ultimately for 76 genes that they believe are medically actionable, return those results to their patients. And some of these 27 diseases encoded by these 76 genes include things like hereditary breast and ovarian cancer, Lynch syndrome, colon cancer, hypercholesterolemia, cardiomyopathy, and the like. And they now have an active program in which they call the Geisinger Genome First program, in which they are returning these results. So studies have shown, our studies have shown that about 3.5% of all study participants in a particular healthcare system that get their exome sequence will have an actionable result. So 3.5% of 50,000 individuals that were sequenced, that's the number that they are committed to engaging and returning these results. I'll point out that, if you extend this to pharmacogenetic results, all patients have a clinically relevant pharmacogenetic result. So this is kind of, if you get an exome in your healthcare system, this is what we're thinking about in terms of downstream requirements, in terms of the workforce and what might need to be done to actually implement genomic medicine. And this paper just came out not long ago, highlighting the first 500 or so patients in which return of results were performed. You can see the disorders here, a lot of BRCA, as you can imagine, a lot of familial hypercholesterolemia, arrhythmias here. This is continued on the next slide of additional actionable results. And you can see a total of over 500 individuals who got their genetic information and for which appropriate follow-up care has been or is being provided. But this doesn't get real until you think about individual patients. And if you go onto the Geisinger website, there are multiple testimonies of actual patients who have had actual benefits from participating in this exercise. So here's Barbara Barnes, a 57-year-old grandmother. She was bringing up 3 grandchildren at the time, found to have a pathogenic mutation in BRCA1. Her response after genetic counseling and the like was, okay, so what do I do next? I have 15 more years to go until my grandchildren are raised. She had genetic counseling and a workup. You can see she had a negative mammogram. She elected to have preventive bilateral salpingo-oophorectomy. At surgery, a stage 1 cancer was found in one of her fallopian tubes. It was removed. She's has gotten chemotherapy and is expected to have a fantastic outcome. My GYN colleagues tell me that the natural history for this would have been that Barbara would have presented maybe in her early or mid-60s with an irresectable stage 4 ovarian cancer, essentially a death sentence. But with participation here and proactive intervention, a lifesaver. And the last one, again, many of these are on the website. Kim, a 66-year-old Geisinger employee, tested positive for Lynch syndrome. She obtained genetic counseling, and her intervention is to get more frequent colonoscopies, and 2 children in their 20s will be screened. So to conclude, I've covered a lot of ground in a short period of time, a lot of little bites of different things, but again, the theme was here to show you how discovery, translational medicine ultimately can result in real improvements in care for actual patients. And these bullets, drug discovery and precision medicine is going to be fueled by human genomics, genetic experiments of nature, can inform therapeutic discovery, and ultimately partnerships to do big science and big data science is where this is all heading, ultimately to accelerate the implementation of precision medicine. So with that, I'll conclude. Obviously, huge numbers of individuals involved with this research at Regeneron, as well as collaborators from all over, and in particular, great thanks to my University of Maryland colleagues for all of the fantastic work that we've done together over the years. Thanks for your attention.
On behalf of the Department of Medicine and IBTA, thank you. Fantastic talk. Question, comments? Andy.
Great presentation, as always, Alan.
Thanks, Andy.
I might add a little bit to that. It was Alan who started the Genomics program in collaboration with Geisinger 15 years ago as part of an office. And you actually brought them into the fold in that project. And certainly, I had no idea that it would benefit us, except that it added a lot of work going up to Geisinger and figuring out how we were going to incorporate them. But you, with Chris Cott and a number of people up there who have since left, Chris is still there, really developed a wonderful program, and you'd be congratulated for it. I was particularly interested in the angiopoietin 4 story, and we followed that up looking at, and I might give some insight into the diabetes risk in the people with low LPL, because we found that the metabolically healthy obese people had higher LPL, higher subcutaneous adipose tissue compared to those with visceral obesity, who were the so-called apple-shaped people. So they did not store their fat subcutaneously. The difficulty with the metabolically healthy people who want to lose weight is that when they lose weight, their LPL goes up even higher, and they regain the weight very rapidly. And we showed that in studies which were published in obesity research in the American Journal of Physiology, that having it puts you at a disadvantage in terms of being obese, but puts you at an advantage from the point of diabetes and cardiovascular risk.
Great. Well, so that's great. It makes a lot of sense. And as all of you know, I love genetics, but at the end of the day, it's all about understanding biology and the underlying physiology. So everything you said makes a lot of sense. And typically, you know, when I see converging lines of evidence, each independent of one another, you tend to believe the story. And the genetics is there, and then also your great studies in the physiology.
We have the DNA from those women in the freezer. I'm not sure, but Alex and Monica were collaborators on that study, and we may have the DNA and be able to sort that.
Okay, I'll be leaving on the Amtrak about 5:30, so get the same.
I know how to find you, so that's not a problem.
Any other questions, comments? So, Alan, let me ask you a question. So you've got the really elegant and solid genomic data, and then you've got the rather less robust EHR data. What are you going to do to solve that?
Yeah, so, you know, the issue of the electronic health record is it's a real-world situation, and there are all sorts of motivations for why certain physicians use one ICD-9 or 10 code versus another, et cetera. So it's a very messy database, there's no question. Even in the best of hands, you know, you can extract data, but it's not the cleanest of data. But the point being here is that what you're hoping is that the very large sample sizes that you can muster from large healthcare systems, and even, you know, collaboration among many healthcare systems, that the signal, that the noise will be drowned out by the signal. And that's the whole philosophy behind trying to harness real-world electronic health record data for these kinds of studies.
Well, let's thank Dr. Shuldiner again.
What's your law? What's your law?